Hello and welcome. I'm just going to double check my mic is working. Yes, it is. Perfect. Hello to anyone out there. My name is Sam and I am an X-ray crystallographer. I have been working as part of the coronavirus structural task force over the course of the pandemic. And what we've been doing is we have been looking at structural data that's been coming out of any COVID-related proteins, and we've been pretty much double-checking that everything makes sense, everything's been processed to the best possible quality. And I'm just going to give you a quick walkthrough of the sort of stuff we do with a structure and why that might be interesting and why it's going to be helpful in the fight against COVID-19. So this here is a structure of the main protease of SARS-CoV-2. So as part of the life cycle of SARS-CoV-2, it produces a very long polyprotein. This polyprotein makes up all of the non-structural proteins that SARS-CoV-2 needs to replicate itself. And it needs this main protease to come along and chop up that polyprotein to release the individual proteins so that it can reproduce. So, in theory, if we can design a drug that stops this main protease from working, then we make it significantly harder for SARS-CoV-2 to replicate. So this is useful in attempting to produce an antiviral. And yeah, I'm just going to show you what we do with the structural data. So this kind of blue mesh that you see here, this is actually our data. It is electron density. So what we do is we take a protein we're interested in, we purify it, and then we crystallize it. And once we've got a crystal, we can fire X-rays at our crystal. This produces a diffraction pattern, which is effectively an array of spots. And these spots give us, we can use these spots to calculate this electron density, and then we can build our model into the electron density. So our model is this yellow array of sticks and such. So this, like I said, is the main protease, and it's an absolutely fresh structure of this. I haven't done any editing to it. So you can see we've got lots of blue density, which means the model agrees with the density. So we've got these amino acids in here that you can see. But then there's also these green bits of density. So what the green bits of density tell me is that something should be here but currently isn't. So most of our proteins are in fact, or our protein crystals, I should say, are in fact mostly water, or mostly solvent. So not the type of crystal you would think about when you think of, say, a diamond or a ruby or a sapphire. These are much more fragile, easily broken, because they are mostly water, and they also can dry out and just fall apart. So yeah, the crystals are very pretty, but yeah, not quite as hardy as what most people would think when they think of a crystal. So one of the major things we have to do when we first start looking at our new structure in its map is we look at these difference density maps. So this is difference density. It's saying, like I said, something should be there when it isn't. You can also get red difference density, where something is there in the model when it shouldn't be. So I can use this to generate a list of all of the different SMAP peaks, and then I can start working my way through these to add in stuff that should be there. So this to me looks like it's probably a water, so I'm just going to add in a water molecule. And then normally we would work our way through all of this, adding lots of waters and stuff as we go. And hopefully what we will find eventually is a very large blob like this. So prior to collecting data from this crystal, what we've done is we have soaked in a sort of a precursor to a drug. So a big sort of influential part of crystallography is the use for structure-based drug design. So essentially we have our protein structure, and if we can then take a precursor drug and soak it in, we can see how the drug interacts with our protein, and then we can use that information to better design our drugs. So I've actually processed this data before. I'm just using it as a very nice example because it has this very strong ligand binding. So I'm going to flip over to my spreadsheet, which is a long list of all of the different compounds that we've soaked into these crystals to try and see how they bind. I'm going to take this smiles string and I'm going to open up my ligand builder. So I'm going to import my precursor drug molecule. There it is. And this always takes a little while to think about. I'm going to add it into my model, and Coot is going to start thinking very hard about this, and eventually we will see it appear in our window here. It normally likes to pretend it's not responding, but it's just thinking really, really hard. So the reason it's thinking very hard about this is it's generating restraints. So this allows us to work with the ligand once we've got it in here, so we can keep our sort of bond lengths and bond angles sensible, and it won't just kind of fly apart into random atoms. So hopefully within the next minute or so we will see our ligand appearing, and then the fun begins as you get to watch me try and fit it into this kind of random blob. You kind of see the blob there, and if you're just looking at this ligand, like, it's not immediately obvious how this is going to fit in, I would say. So obviously I have an advantage that I've already worked with these data, so I do know how this is built, but it can be a bit of a challenge if you're working with, especially with data which don't necessarily have a very, very strong difference density, to try and build these ligands in. It can be quite challenging. But hopefully soon this will be ready, and you can just watch me fight with it pretty much. If anyone out there does have any questions, do feel free to put it in the chat so I can attempt to answer. I will note I'm obviously not a medical doctor, so I won't be offering any medical advice on COVID-19, but I can tell you about structural biology if that's something that might interest you. Yeah, I have a passing understanding of all the vaccine stuff that's been happening, but again, not really my field. Come on, ligand. And Any second now. There we go. Okay, we've got our ligand in. So I'm just going to get rid of this so you can see it a bit better. So what I'm going to do is I'm going to go up here to map, and I'm going to switch to this weighted difference map from refinement. So this just means I'm going to pay attention to the green map instead of the blue map. And what I'm going to do, if I click this button up here, the real space refinement zone, and I click on my ligand, it's going to start wiggling, and it's going to try and fit into this green difference density. So currently it's kind of wiggling the wrong way. So what I can do is I can guide it to where I think it should be. So as I said, I've already worked with this structure before, so I do know how this should look. So I know this carbon should be down here, and it will be covalently bonded actually to this sulfur here on the end of this cysteine. It's not wanting to cooperate right now. Okay, so we've got that just about in the right place, but obviously all of this stuff over here, completely wrong place. And we need to try and drag this around here so it picks up this bit of density. And we want to drag this up here. And now, hopefully, it's going to start settling into a nice position. It's going down here a little bit. Okay, that's making its way. But this is obviously still not in the right position, so we want to try and flip this 5-membered ring. If we can get hold of it. No, that's the wrong one. Come on. No, I don't want you. Flip. Okay, so we flipped that, and now that means this can settle into the right position quite nicely. And then if we look up here, you can see that the density isn't perfect, but we have a fairly convincing fit. And if we look on this panel here on the right, you can see our bond lengths and our bond angles all make sense. So what we're going to do is accept that. That's going to stop jiggling about, and it's going to stay there. And now what we're going to do is we're just going to merge our molecules together, like so. So you can see now it's turned yellow instead of green, just like our other model. And then what I'm going to do is I'm going to save that molecule. Press OK. I'll close down Coot and come back into CCP4 here. And now what I'm going to do is I'm just going to run a quick refinement, and while I'm at that, I'm just going to quickly turn off Teams. So I'll be back. Okay, we're back, and refmac is currently running. So what refmac is doing right here, go away Teams. What refmac is doing is effectively recalculating our electron density based on the model we've provided. So now that that ligand is in place, it will recalculate this map, and if all goes well, the previously green density, where we've now put our ligand, should turn blue. And that will tell us that we have, in fact, modeled our ligand correctly, and that it is in the right place. So hopefully refmac will finish soon, and we can open up Coot again and have a look. Okay, so I'm going to click Coot, tell it to run, and it should appear soon. Okay, we've got Coot. So, quite a lot for my laptop to handle, really, with streaming software and data processing. It's a poorly treated laptop, I'm afraid. But yeah, if we flip over here, okay, so we can see it's turned mostly blue, but we've got a little bit of red going on. So what could be happening is we don't actually have 100% occupancy here of our ligand. So our data are made up of, the data comes from a crystal, so it's actually a sort of average, really, of all the molecules that make up the crystal. And if, say, 50% of our molecules in the crystal have the ligand and 50% don't, then the final result is a composite of both of those data. So half of it's going to be saying it's there and half of it's going to be saying it's not. So what we can do is we can go to residue info here. We click on our ligand and we can change our occupancy to sort of 50%, 70%, 60%, whatever we think works best, and then we'll run it back through refinement and see if it just comes out blue or green if we've gone too far either way. But yeah, that's enough about the ligand. Let's show you the other sort of things we look at in our protein structures. So this here is called the ramachandran plot. And what it tells us is if our secondary structure makes sense. So secondary structure is beta sheets and alpha helices. We do have an article up on insidecorona.net explaining about secondary structure and protein structure in general. But just very quickly, I will go through some of it. So this here, this region up here on the top left, is beta sheet regions. And what you can see these kind of two parallel, with three parallel lines of amino acids, and they're interacting by these nitrogens and oxygens. You can see another one here: oxygen, nitrogen, nitrogen, oxygen, oxygen, nitrogen, to form this sheet-like structure. And our Ramachandran plot gives us an idea of if our secondary structure sort of falls within the realms of possibility. So if I click on this red one here, this one's an outlier. So it doesn't quite fit with what we know is the structure of a beta sheet. So what I can do is I can turn on my Ramachandran restraints, change the weight a little bit so that Coot is going to pay more attention to bond angles and lengths instead of my electron density. And I can just refine this whole area with the hope of improving my Ramachandran plot. So you can see here we've got these spheres that have appeared, and if I move this, you can see them changing color as I'm putting it into a bad position and good position and everything in between. So that seems to have settled into a good position. Like I mentioned before, we don't always have perfect density around our amino acids, so this residue here is obviously very, very flexible. And the problem with very flexible residues is it's really hard to pin them down into a single place. So this is probably just flopping about all over the place. And that's why we haven't got nice convincing structural density anywhere. So yeah, but what we're going to do is we're going to accept that. and you can see our red point has now moved into the favored region. The second type of secondary structure is an alpha helix, which you can see here. You can kind of see this coiling around, and again, this is governed by nitrogen and oxygen interactions. You can see here. And yeah, this also has sort of defined angles that we know typically make up an alpha helix. So if we look at this outlier here, so this is still obviously making a helical shape, but our bond angles are just maybe a little bit off. So we're just going to do the same thing again. I'm going to do a quick sphere refine, and yeah, you can see that's managed to sort of draw everything into the right place. This was obviously not very far out of an ideal range, so it hasn't really changed a lot as far as we can see, but it just means the bond angles are a bit more what we would expect. Okay, so yeah, we check for ligands we want to add, we go through and make sure that our secondary structure makes sense. There's lots of other stuff we can check as well, so if I fire up the rotamer analysis, let's have a look at some of these bad rotamers. So this happens a lot with these very, very flexible residues, that it's difficult to, there's a lot of ways they can be arranged, but we have sort of a list of conformations which the bond angles and lengths make sense. So we can flick through these and try and find one that fits better to the density that we've got. You can't always do this. Sometimes things adapt non-optimal conformers, and you just have to accept it. The other thing you can do is you can try and force this into a more accepted rotamer by using this auto fit option. But as you can see down here, this has really messed with the backbone. So I think if we try and straighten out our backbone, it's probably going to just come right back as a bad rotamer. So yeah, we kind of just have to accept this and move on, really, because it's much more important that this backbone makes sense than this really, really flexible residue is defined. Yeah, that is probably the bulk of what we're doing when we're going through the structures. We can also check all of the water structure, but there aren't really many waters in here because I haven't added any yet. So yeah, that is, for the most part, what we are looking at in our structures. Once we've gone through this process quite a few times, so we'll add stuff here, we'll run it through refinement, and then we'll open it back up and check that it makes sense, add more stuff, take stuff away. It's a very iterative process, but eventually we end up with a structure that we are happy with, and then we will load it into PyMOL, which is this program here. And PyMOL pretty much lets us make pretty figures. So I'm just going to change the background to white. Here is our structure. So this is a lot sort of easier representation to see of beta sheets and alpha helices. You can see here. We can do lots of stuff in PyMOL. We can color it in however we want. It's an American spelling. So I'm going to color aquamarine, my secondary structure, and I'm going to make my helixes blue. And then I'll color in forest, the beta sheets. There we go. And let's color in wheat, the secondary structure of loops and everything else. So now you can get sort of a much clearer view of the different regions in the structure. You can also then color by element. Personally, I like to set my helices to fancy. I just think it looks nicer. Personal preference, obviously. But yeah, so this is the sort of software we'll be using to make our pictures. Now, obviously, really, a protein doesn't actually look like this with the arrows and the helixes. What it really looks like is kind of a blob. So I'm just going to switch this over to a surface view. So there you go. You can see this is what our protein actually looks like, and you can get a pretty good view here. Center on our precursor drug. You can see how it kind of fits nicely into this pocket. So if we were trying to further design this drug, you can kind of see we've got this pocket down here where we could definitely build something into, and that will hopefully improve the binding of our drug. And we'll end up with a more specific and a more sort of powerful treatment. The other thing we can see, I'm just gonna, let's bring back the cartoon. Where's cartoon? There we go. The other thing you can do, I'm just gonna hide those ribbons. There we go. We can generate symmetry mates. So obviously this is, like I said, it's made up of a crystal. And we actually know that the main protease works as a dimer. So two of the same protein bind together to form an active protein that can actually do its function. And we can recreate that dimer if we can find the right symmetry-related molecule. So we'll just flick through these and hopefully we will find the one related. There we go, that's the one. So we'll just turn off these other ones. And there we go, there is our full protein. And I think the ligand has disappeared, so let's just show that again as sticks. So probably a good bet for stopping this protein working is to be able to stop it forming a dimer. So if we can, from our structure, maybe figure out a way to bind into this region and prevent this dimer forming, then the protease isn't going to work. The other thing we can do is we can try and clog up the active site where our protease would normally cut stuff. Yeah, that's pretty much it, really. So we can show surfaces again of both of these. You can see how they interact. This is a case where it might actually be more clear to change our colors to separate ones. You can kind of see here how it all binds together, and actually our drug isn't actually too far away from the dimer interface. So if we could grow this drug in this direction towards the purple, then maybe we can stop this dimer forming. But yeah, that's pretty much all I have to say on this. The only other thing would be check out insidecorona.net. I will corona.net. So this is the website of the Coronavirus Structural Task Force. We've got lots of stuff up for the general public, information about the coronavirus, how it enters the cells and all that sort of stuff. Lots of different articles about sort of the basics of structural biology, and also some more specific stuff about the coronavirus. We've got lists of all of the proteins that have been solved so far, as well as a resource for the general public. This is aimed at people that use structures further downstream to sort of do simulations and try and design drugs, which they then feed to a chemistry lab, which would make the drug, and then the drug would probably come back to structural biologists, where we would, again, soak that drug back into the protein, see how it binds, and kind of go from there. So yeah, thank you for watching. I hope this has been informative, and yeah, I'll see you again next time.